Hello everyone, we will be presenting the computational platform for synthetic correction of disease-causing SNPs by Luke, Alexander, Minh, and Heewon. So first we'll be presenting our goals and motivations. Our motivation is to create a website that can identify SNPs and create guide personalized genetic correction strategies for the users. So our main goal is to make a website that identifies disease-associated SNPs from patient sequences and generate CRISPR based on correction strategies and evaluate gRNA based on the efficiency and off-target safety. So our general functionality workflow, as shown here, explains that there's an input where the user can input and paste a sequence or input a FASTA or a TXT file. Then the analysis begins. When the SNP is detected, it will show the SNP number, SNP position, reference, and suggested nucleotide. Then the exported results would be in FASTA file, which could be exported and used on a different third-party website, just such as Benchling. Or we have another option where in a inputted embedded function for gRNA and CRISPR design is within the website.
Our design specifications focused on accuracy, safety, and usability. The system accepts FASTA files or manual DNA input with a minimum query length of 30 base pairs, while rejecting malformed or duplicate database entries. It is designed to detect SNP positions accurately, and its gRNA scoring considers PM presence, distance to the SNP, GC content, homopolymer penalty, and off-target activity. Finally, the GUI was built so users can complete the workflow clearly in 7 steps or fewer.
In a more abstract detail, the workflow is organized into integrated layers. BLAST code PY is a backend helper file for input handling, FASTA formatting, database management, BLAST execution, and output parsing. The main issue it solves is that BLAST needs clean DNA input and proper FASTA files, so the code adds safety checks before anything reaches BLAST. Normalized sequence cleans any DNA sequence-related input by removing white space, converting it to uppercase, rejecting empty input, and blocking invalid characters outside A, C, G, T, and N. For database safety, merge FASTA records calls deduplicate records to remove repeated sequences before rebuilding the database. Finally, run BLASTN calls BLASTN executable file through run command using subprocess.run.
This demonstration uses a mock GUI that shows the intermediate backend steps more clearly than the final GUI, including the input safety checks and database handling. The BLAST bin folder is already configured. First, I manually enter a sequence with an invalid nucleotide, and the system catches the error instead of storing it. Next, I enter a sequence that already exists in the database with a different header. The system detects the duplicate sequence and prevents it from being added again. After confirming the safety checks work, I ran BLAST using the query sequence. The GUI then displays the parse alignment output, including the match sequence header and alignment metrics.
The SNP analysis Python file turns BLAST alignment output into SNP results. The core detection happens in compare aligned sequences, which uses the BLAST start and end positions to compare the query and reference base by base. Any mismatch is recorded as a possible SNP with its position and nucleotide change. The main function, analyze with disease FASTA, connects the full pipeline and builds the report rows. After BLAST completes the alignment, the app displays metrics such as percent identity, mismatch count, and bit score, which will be used for best SNP compatible BLAST alignment. Before running the SNP analysis, the disease database provides the known SNP reference. When we click Show SNP Analysis, the backend calls SNP analysis PY. It compares the patient query sequence against the matched reference sequence to detect the mutation nucleotide. The detected mutation is checked against the disease database. If the position and nucleotide change match, the output shows the SNP position in both query and reference, the nucleotide change, disease association, and pathogenicity. This information can be used later for gRNA generation.
The CRISPR logic file's role is to identify and rank optimal gRNA sequences near a mutation site for CRISPR gene editing. The scoring system itself combines these two biological factors into a composite score. However, an important limitation is that these scoring weights are heuristic and have not been experimentally validated or trained on real CRISPR efficiency datasets. This means the ranking is useful for prioritization, but may not perfectly reflect real biological efficiency. The main function, find gRNA candidates, brings everything together. It scans both DNA strands for NGG PAM sites, extracts 20 base guide RNA candidates, scores them, and returns the top-ranked results.
The front end is the React single-page application. Every user action that needs computation, such as sequence alignment, gRNA design, binding site lookup, will fire an HTTP request to a fast API backend, and the backend calls BLAST executable and sends structured JSON back. In the Python backend, we set up fast API app with cores to act as an entry point for Unicorn, and we also implemented some GET and POST routes for the backend APIs, which includes the full BLAST and SNP pipeline, the metadata sidecar, BLAST database, and CRISPR design. Here's the code to show the global state and the workflow of our app. The main analysis is a linear pipeline from sequence, SNPs, gRNA design, and binding sites. Each will have its own page, and instead of passing props through every level of making duplicate API calls, we store the pipeline state in a React context. Here's a demo of our web app. As a new user, you will need to go to Setting and set up the BLAST bin folder and the database. The instruction to do so can be found in the Instruction tab here. I already have my BLAST bin and database set up, so now I can go to Analysis and input my sequence, which is a human HBB mutated sequence. And when I hit Start Analysis, it will give me the result, which in this case, it found one SNP at position 396 corresponding to sickle cell disease. Clicking on it will give us a collection of guides used for CRISPR design and the corresponding score. And we can also view the genomic binding site of the guides and our input sequence. And after we finish viewing it, we can go back and export the result in any form we want.
This is the error example of the gRNA design visualization. The yellow box highlights an incorrect SNP nucleotide call, mainly caused by an indexing error. The red box shows two additional issues: an incorrect complementary sequence and a cut set that was much farther from the SNP than expected of 4 nucleotides away. Our key successes were reliable input validation, correct SNP position detection, full integration of the Python modules into one GUI, and a clear workflow that users could complete in fewer than 7 steps.
Our main limitations were BLAST alignment ambiguity, database metadata dependency, and incomplete calibration of the self-coded gRNA module. These issues caused SNP analysis errors, unknown variant matches, and unreliable guide ranking. So future work will focus on better filtering, stronger database validation, and benchmarking against gold standard tools.